Now look at this.
What's up, Big Bad Penguins? Today we're going to do topic 3.3, environmental impacts on enzyme function. Enzymes are proteins. So the different things we've talked about that are going to denature a protein are going to denature an enzyme. So we have these two different enzymes. You have your pepsin and you have your trypsin. And so you can see that the rate of reaction kind of goes down when it gets away from their optimal range. That's because if you change the pH, it's going to denature your protein. It's going to cause that protein to unravel, and it's no longer going to have that active site for the substrate to bind to it. Excessive heat can also cause a protein to denature. And so if you find that you have an enzyme and you apply too much heat to it, it can denature it, which is going to decrease your reaction rate. Three different things can happen. One, you could have no effect because the active site is still functional. Two, we could have reduced function because now the substrate is unable to bind to the active site, so it can't speed up a reaction. Or maybe you could have a new effect. So there could be now something else that can bind to it, and now that protein can cause some other reaction to take place. So when you denature the protein, sometimes it's reversible, as in it'll be able to fold again. There's a protein inside of Jell-O that does this. Sometimes it's irreversible. So once you denature the protein, it's not going to be able to reform. Example being the albumin that's in the egg whites. So when you crack the egg, it's colorless, and then once you apply heat, it becomes white. So let's go back to pH and how pH is going to cause the protein to denature. So pH, the solvent, is the negative log of the concentration of hydronium. So oftentimes we're just going to see this as the exponent. So for example, if we look at a pH of 3, it's 1 times 10 to the negative 3 versus a pH of 6 is 1 times 10 to the negative 6. And so as your pH decreases, your hydrogen ion concentration increases. As your pH increases, your hydrogen ion concentration decreases. So they're kind of like inverse in a way. The reactants in the product concentration is a big important thing. So if I have a high concentration of reactants and a low concentration of product, there's going to be a higher chance of the reactant to bind to the enzyme, so we see an increase in the reaction rate until we reach saturation. If there's a high concentration of products, that's a problem because the product can bind to the enzyme, which can decrease the reaction rate because it causes product inhibition. So speaking about inhibitors, what's the difference between competitive and non-competitive inhibitors? So traditionally, substrate is going to bind to active site, right? So competitive inhibitor is going to bind to that same active site to compete for the active sites. That's why we call it a competitive inhibitor. Non-competitive inhibitor is going to bind to a allosteric site or another site on the enzyme. And so when it does that, when you bind to a protein, it changes shape. So the change of shape inhibits the ability for our substrate to bind. So inhibitors are going to decrease the reaction rate. Want to overcome a competitive inhibitor? Increase the reactant concentration. Increased temperature causes an increase in the kinetic energy of the molecules, which is going to cause denaturation. A decrease in temperature is going to cause a decrease in the kinetic energy, which is just going to slow down.